Hello and welcome to this webinar titled Do Extractable Protocols Truly Help? An End-User Perspective, hosted by BioPharma Asia magazine and presented by Ekta Mahajan, Genentech Roche, and Dr. Saskia Haehn, Laboratory Manager for Extractables and Leachables at Central Analytics of Merck KGaA Darmstadt, Germany. My name is Stephen Edwards and I'll be your moderator. Now please allow me to introduce our first presenter. Ekta Mahajan is a Principal Engineer in Pharmaceutical Technical Development Engineering Group at Genentech Roche in South San Francisco. In her 14 years of research and manufacturing experience in biotechnology, vaccines, and pharmaceuticals, she has led various projects in a wide range of areas, both upstream and downstream, including commercial manufacturing support, process development, and new technology evaluations. Ekta has experience in disposable technology and is currently leading projects for design and implementation of disposables in development and manufacturing, including drug conjugates. She is an active member of BioPhorum Operations Group, ASTM, working group, and recently successfully organized the first science-focused single-use international conference through Engineering Conference International. In October 2015 and May 2017, which is being converted into a series. Before Genentech, she worked as a staff/senior engineer in the technical operations and engineering and the technology departments at Merck KGaA Darmstadt, Germany. Ekta has a BS in chemical engineering from the Thabta Institute of Engineering and Technology in Punjab, India, and an MS in chemical engineering from Bucknell University. I will now be handing over to our first presenter. Welcome, Ekta.
Thank you. Hi. So I'm going to talk about the BPOG Extractable Protocol truly help. It's something I think that has been going on for a long time. So basically, plastics, right? It's not something that's new. We have been using plastics for a very long time. We see it in our day-to-day applications. We have plastic bottles. There's food industry heavily uses plastics. It's been used in medical devices. So what is a plastic and why do we worry so much about it? So they are typically organic polymers, right? Majority of the plastic is formed with polymers, but then you have additional stabilizers and fluids that are added to help with the process or increase the durability of the plastics for various reasons. But one of the drawbacks of adding all these additional components is that they do lead to extractable and leachables. The fact that they have been used in the industry for so long, why is it that we still continue to worry about the plastics? Why do we care so much about it? And the reason is we are now using them for pharma industries. It's very different when you're using it for in a food industry where you're ingesting versus when you start using in biotech where you are now actually injecting into patients. And also you're dealing with cells that can be sensitive. You don't want to add anything additional that could impact the cell growth. You want to make sure that the drug product that you have manufactured is stable. There's nothing that will interact with our proteins that could go into patients and could cause any adverse effects. So the patient safety is the utmost important thing for a biopharma company. So if we are making drugs to save people's lives, not cause any additional adverse events. Because of that, when you're doing the process, so you need to make sure that whatever material that you're using is suitable for a process, whether it's temperature or physical characteristics. You want to make sure that you're not going to have any leaks. It's able to maintain or be able to suitably make your product without any issues in the process. Because if you're not able to manufacture your product, you won't have anything to supply to your patient. So the supply chain is always an important aspect. You want to make sure that you never shortage for your patients. And then lastly, we need to make sure that we understand what is the impact if any changes are made by the supplier. Again, kind of tying back to all the things that I mentioned before. If there are any changes that are made, how does it impact the cell culture, protein stability, patient safety, and making sure that we continue to work with the suppliers to understand the impact, and they understand where, how the changes that they make could impact the processes that the applications that we're using them for. So how do we address these challenges? We can continue to work one-on-one with the suppliers, which can be very intensive. And of course, when you're doing that, you also get to hear how you guys are the only ones asking for it. But in reality, every end user is asking for the same information. They are dealing with the same challenges. So you could partner as an industry group and address the challenges together. And you can have the information sharing. It's better for the industry, it's better for the patients. Again, the end goal is always patient safety and patients, right? So the more you can work together to make sure that we are delivering products to the patients and saving lives is always the best thing. So as part of the industry group, BPOG is one of those industry groups which came together. It has almost 24, 25 companies across the globe, which came together because each one of them were having the same problems. They wanted to work towards a common solution so that we are not impacting the application of single use, or worse, we are not doing anything that could impact the patients. So when the BPOG group came together, the first goal was related to the Extractable Protocol, was how do we develop a standard protocol for testing single-use components? And why do we want to do that? One of the challenges when you don't have a good data set, it takes a long time to qualify a single-use component because you have to work with the labs to generate data. But if you get the right level of information from the suppliers to begin with, you can reduce your qualification to 4 to 8 weeks instead of a year. The protocol should meet majority of the biopharma needs. The rationale behind that was the 80-20 rule. There's always will be unique conditions for every company that they'll have to do some extra work. But the idea was when you're working on a standardized protocol, it has to meet majority needs, not each and every need. It helps with troubleshooting or resolution of issues when you have problems. If there is data, you can start looking at, okay, what caused my cell culture growth, or what caused my unique peaks coming in my stability pools, right? But if you don't have data, then you have to start from the scratch, which again is sort of always a luxury when you're working in a manufacturing environment where you are trying to make product for the patients. It helps when you have, again, when you're looking at your products, you're trying to see what is the best option that will let me continue to manufacture without impacting my patient supply to the patients, right? So you want to make sure you are able to get the best product which has the least extractables in it, so that you can, you're not, again, putting anything into drugs that could impact the patient. And then the Leachable studies, as we get closer and closer to the drug product, you have to have Leachable studies because you want to make sure, again, from the patient stability perspective, that you're not putting anything in there that can cause adverse event. So the Leachable studies have to be done, but they need to be established, or the extractable data helps with understanding and looking at what could be possible in a Leachable study. So when we do our stability studies, it helps when you are establishing your product expiry and making sure that your product is going to be stable. So how did it proceed. So initially, as you can see, it's been a long time since this discussion has been going on. So back in 2012 is when I published a paper along with colleagues Trishna and Jim Logo in an ISPE. As part of ISPE, we had solved or having worked on qualifying single-use components was something that we saw as a problem, and so we published that paper in the hopes of forming an industry group, and we got the good response. We did establish a BPOG group back in 2013, and then along with the end users, we came up with a draft, which was then discussed with the suppliers one-on-one in a group setting with BPSA, with ASTM, and based on that feedback, the model was it was modified. The protocol was modified to incorporate the feedback from them. And then after the feedback was incorporated, then we published another protocol in ISP in 2014, and it was shared with the companies for incorporation. So there was a lot of work that went into in discussions with ASTM, with the USP. So within the companies, the protocol has been incorporated. There are some suppliers who have generated data and some suppliers who still continue to drag their feet, unfortunately. So, personally, from the end user perspective, the data from the BPOG protocol definitely helps with the qualification process as well as troubleshooting. When you're working in the manufacturing setting, you constantly have demands or questions that you need to answer in a day or two, and not having that data doesn't help. So these were the conditions that were established as part of that protocol, where we looked at all the single-use components used in the processes end to end, and looked at what are the suppliers, what are the solution, what are the bracketing, what kind of solutions that get used in biopharma, and so the solutions were selected based on that, based on the pH, based on the polarity, and then the time points and the temperature were selected based on, okay, what's the longest duration that a bag could be used, or a filter or a tubing can be used, and then we selected different time points based on that. And the reason for selecting different time points instead of just one time point is because you have volatiles, non-volatiles, semi-volatiles, and they behave differently over time. So just having the last time point does not give you enough information, because you get information only from non-volatiles, and you would have lost all the other information. So what do we do with this extractable data? So this is a case study just to show that, an example to show that, okay, let's say we have a bag, and we look at the surface area, we look at how many ports are there, so that gives us an idea of the total area. And then we have a product that is going to go into this bag, so we know the highest expected grams for that product. What is the daily dose? What is the patient average patient weight that is used for these calculations? So then we calculate the estimated daily intake based on looking at the extractable mass per race, multiplied by the surface area, and then daily dose per product mass. So this is then compared to acceptable daily intake, and if the estimated daily intake is less than the acceptable daily intake, then it's considered to be safe. But if it is same or more than the daily acceptable daily dose, that means that that product cannot be used. So now moving on to the case study. So we had a single use was going to use for project A, and the goal, like I said, a lot of times in the projects you don't really have too much time. So that group came to us saying they wanted to qualify the probe for use within 1 month. They have timelines. They wanted to use single use, but can they use it? Looking at if the solvents would fall within the model solvents or not. The solvents that are going to be used for the process will fall within the model solvents or not. So the next step was then, okay, does the supplier have that supporting extractable data? Because qualifying within 1 month is not feasible if we don't have the data. So fortunately, in that case, when we approached the supplier, they did have the BPOG extractable results. We performed our tox calculations. We found that there was no class 1 solvents as per the ICH-Q3C guidelines. There were 2 class 2 solvents that were detected with 50% ethanol and 1% polysorbate, and then 1 class 2 was detected in backport. But when we looked at the permitted daily exposure, we found that for all these cases, the estimated daily exposure was less than the permitted daily exposure for all these 3 extractants. So we felt that it was safe to continue using them. And then we did the summation of all the known extractables. So because of the new solvents and new time points that were suggested as part of this protocol, it was expected that there would be some unknowns as the suppliers and the labs continue to come up to speed on analyzing these new compounds. But for our purposes and for the tox calculation, we did have to do, look at the known and the unknown. So for the known, it's easy because then you can compare it to the permitted daily exposure. But when you have unknowns, you cannot do that. So you take the worst case scenario and you assume that everything that's unknown is toxic. And then you compare it to the toxicology threshold of toxicology concern, which is the TDC value. In our case, the estimated daily exposure, assuming it to be worst case, was still below the TDC. So it was considered to be okay. And then we looked at the leachables from 20% DMSO because it was the concentration of DMSO was beyond the model solvent bracket approach. So we did studies with 20% DMSO. But again, we did not find any class 1 and 2 solvents, and the other extractants that were detected were below the acceptable daily exposure, so film was considered suitable for use. So overall, summarizing it, the availability of data definitely supported the project timelines. Like I said, within 1 month, without data, it would not have been possible. The extractables were within the extractable limit, and film was tested suitable for use for the intended purposes. So next case study where we needed leachable assessment for drug substance and drug product bags and connections. Again, the research from the team was to perform the leachable assessment for bags along with assemblies in 2 months. The system had connectors, bags, tubing. So I'm not going to repeat what, I showed how we do the calculations for the first case study. So I'm just going to show whether we had information or not for the next 2 case studies. So in this case, the leachable assessment required extractable data, and the extractable data from the suppliers was available, and it enabled execution of leachable studies to meet the project timeline. All the leachables were within acceptable limit, and then if we had not received the data from the suppliers, appropriate data from the suppliers, we wouldn't have been able to meet our required timeline, and it would have taken us 1 year, which would have resulted in the project team using stainless steel instead of single use, or we would have had to delay the project. And most likely they would have gone with the stainless steel option because delaying the project is never an option. Now, case study 3, where we had to do an extractive assessment for a filling line. Again, we had to do this stress assessment and to enable filling of products in vials using disposable assemblies. In this case, we were not too fortunate. The data from the supplier included only TOC results, and it's impossible to perform tox calculations. And as per the health authority guidances, we do have to list all the extractables and leachables in our submissions when you post UFDF. So not having the data is not, again, an option. We have to either generate. So in this case, since we didn't have the information and then we had to do internal studies to address missing information, it took us 6 months to perform the assessment due to lack of data. It impacted the project resources within deciphering the batch supply data. When I say batch supply data, the data that was provided by the supplier was really, again, did not meet the requirements and had a lot of, it just wasn't the appropriate data. And then it introduced such kind of information, or when you don't have the right information, it introduces the hurdles for implementation of single use, because when you don't have information and it takes a long time, the project teams tend to move towards, going back to the stainless steel. So it definitely is not good for if we are trying to move forward with single use. And then any supplier that does not provide that information does fall from the preferred supplier list. So just to conclude, availability of appropriate extractable data enables execution of projects with short timelines, and the suppliers providing data become preferred suppliers. It lowers assessment time, it's efficient utilization of resources, and then, as I've in last a little over a year, having moved into the regulatory group, now seeing from the other side, it becomes even more critical as I continue to see the number of questions that we get, and the, not just from the technical side, but also from the compliance side. So just to end the talk with acknowledging people that have helped in these different stage studies, or have been part of the assessments. And of course, the BPOG Extractable Leachable team, this would not have been possible without that team. Yes, thank you.
Thank you, Ekta. Once again, I'd like to remind our viewers that there will be a live question and answer session at the end of the webinar. But please feel free to send in your questions at any time during the webinar via the questions tab located directly below your webinar screen, and we'll go through these at the end of the presentation. Now please allow me to introduce our second presenter. Saskia studied organic chemistry at the Saarland University in Germany, with a focus on analytical chemistry, especially gas chromatography, mass spectrometry. After her PhD, she worked for a contract lab as project manager for E&L to support clients from the pharmaceutical and medical industry. In 2014, Saskia joined Merck KGaA Darmstadt, Germany, where she set up the laboratory for extractables and leachables in central analytics. She applies technical and regulatory knowledge, scientific experience, and expert judgment to address solutions for a broad range of difficult problems. In her role, she is responsible for generation of extractables information for improved dossiers. I will now be handing over to our second presenter. Welcome, Dr. Haehn.
Thank you, Stephen, for the introduction. My part of the webinar is also about standardized protocols for generating extractables data. However, the focus is more on the analytical part. In addition to a short introduction to single-use systems and current standardized extractables protocols, I will discuss which extraction solvents have been provided additional information in our studies and the analytical challenges that we have faced with these protocols. The biopharma market was valued as $200 billion in 2016 and is expected to grow with a compound annual growth rate of 15%. The adoption of single-use bioprocessing products had witnessed a significant increase during the developmental stages of drugs and manufacturing of several biopharmaceuticals. This product had revolutionized the upstream as well as downstream processes of manufacturing drugs, making it faster, simpler, and cheaper. In addition to reduced capital investment, the top reasons for the implementation of single-use are the reduced time in getting the facility up and running, the eliminating cleaning requirements, and the reduced risk of product cross-contamination. However, certain aspects, such as extractables and leachables issues and stringent regulation pertaining to use of disposable systems in biomanufacturing industry are restricting the use. Single-use bioprocessing employs disposable technologies for the development and manufacturing of pharmaceutical products such as vaccines, personalized medicine, or others. As an example, a typical monoclonal antibody as well as a CAR T-cell therapy process are shown. Most of the unit operations consist of polymeric components. The safety risk associated with extractables from these components depends on a number of factors and is assessed via application of a risk evaluation matrix. Moving on to standardized protocols. Why try to standardize extractables data at all, or rather the requirements of the procedure for it? The key benefits to do so are highlighted here. Expectations for supplier extractables data are standardized. End users can use the data which are provided by the suppliers to do their risk evaluation and do not have to carry out any or only reduced extractables data themselves, which then significantly reduces the cost and also timelines. And in addition, the selection of the right component is made easier by the comparison of different suppliers. One of the two standardized extractables testing protocols is the so-called BPOG protocol, or BPOG Standardized Extractables Testing for Single-Use Systems in Biomanufacturing, that was published November 2014, and Ekta has presented in the first part of the webinar. This protocol describes very extensively both the extraction in terms of extraction solvents that are shown here in the left box in the middle, extraction conditions and time points in the two boxes below, and the analytical methods to be used for quantification and identification of the extracted entities, including also reporting limit and additional information. The other protocol is described in the USP 665 that is currently in its second round of public comment. Comparing both protocols, USP 665 specifies only 3 rather than 6 extraction solvents: the organic, the low, and the high pH. The organic stream with 50% ethanol is identical in both protocols. The low pH streams are considered interchangeable. With the basic solution, it comes down to the process stream. USP describes one extraction time regardless of the component. The focus of the protocol is clearly on the extraction approach. As far as analytical methods are concerned, reference is made to USP Chapter 1663. The USP Chapter 665 will not become official until December 2025. In a holistic sense, both protocols help the end user. The protocol expectations are slightly different as shown on the two overview slides before. For comparison, the sample schedule for a filter according to BPOG and the USP 665 protocol are presented here. Due to the lower number of extraction solvents, time points to be examined, and batches, the number of samples to be analyzed is reduced compared from BPOG to USP by a factor of 12. Coming from the standardized protocols to how MilliProSigma supports their customers in this topic. The industry-leading Improve program provides documentation and regulatory information on materials used in the manufacture of drug products, facilitating manufacturers' risk assessment workflows and suppliers' qualification, as well as enhancing transparency. This program has expanded 2016 to include a selection of products for filtration and single-use processing. With the Improved program, customers have instant online access to regulatory and technical information on a wide range of products. Depending on the type of dossier, there were different information available. You have the material qualification dossier that provides information to start a qualification. There's the quality management dossier that provides you information to answer questions during your risk assessment. And last but not least, you have the Operation Excellence dossier that supports you to support process optimization. The last dossier is also the one that contains the full Extractables profile. What does this mean in detail? You have the representative sample selection for a product family, a scientific rationale on building a worst-case scenario to the testing strategy, a robust extraction process to maximize potential extractables from the sample, an elaborate analytical process to facilitate the detection, a full-scale set on elemental impurities as per USP 232 and ICH Q3D, and an executive summary of full extractables profile. The improved program report combines the requirement of both the BPOG with the additional caustic solvent stream of the USP 665. In addition to the required instrumental analytic approach, ion chromatography is performed additionally for ions such as halogenides and small acids. We will now change the focus to analytics. We have seen that there are differences in the BPOG and USP protocol in terms of extraction solvents. The key question is which extraction solvents make sense and provide complementary information. The data that are shown here are from existing data we have generated in the past according to BPOG requirements. Similar conditions were used for all the 3 devices. Comparison of the results showed that all substances found in the salt extract were also detected in the water low or high pH stream. Similarly was this observed for polysorbate 80. All substances that were present in this extract were also detected in the 50% ethanol extract. These 2 extraction solvents, the 5 molar sodium chloride and the 1% PS80, have not provided any additional information for these components. Let us proceed from the comparison of extraction solvent to which analytical challenges are there. On this slide, you can see the GC chromatogram of a 1% PS80 controlled sample after liquid-liquid extraction with dichloromethane. The liquid-liquid extraction is a standard sample preparation for water-containing matrices. Besides the BHT, which is spiked to determine the recovery, and the 2 spike standards, the origin of all other peaks is the PS80 matrix. In this example, the concentration of these 28 peaks is above the reporting limit of 100 ppb and reaches up to 2.5 ppm. This high matrix interference makes it quite difficult to evaluate in the interfered spectra, especially in a non-target analysis. Recovery and the stability of the instrument decreases due to matrix effects. The same phenomenon occurs also in liquid chromatography. The figures show the chromatograms for UV, ESI negative, and positive detection. For mass detection, both the TIC, the total ion chromatogram, which is the sum of the signals from all masses in any given spectrum, and the BPC, the base peak chromatogram, that's the signal from the most intensive mass in any given spectrum, are shown. All integrated peaks have a higher concentration than the reporting limit of 100 ppb. In case of co-elution, quantifying and identifying the actual extractables is quite hard. Ion suppression can also occur through the matrix. Besides the general PS80 matrix interference, the degradation of PS80 is an additional challenge. The degradation depends not only on temperature, also on the contact material as it is shown here in this overlay. This phenomenon clearly comes into effect when filling the test material to be examined for extraction with the PS80 solution, whereas method control is stored in a glass or a Teflon container. Due to these different contact materials, a different degradation can take place, and thus components of the matrix could be reported as extractables of the test material. The basic requirement of an extractable study is to be able to identify and quantify substances that have been extracted from a test material. We conducted the following mixed spike experiment to verify this for the 6 well-known extractables. These substances were spiked into the PS80 solution at a concentration of 1 and 10 ppm. The results for ESI positive and negative are shown here as base peak chromatograms. Red is the control, blue a concentration of 1, and black a concentration of 10 ppm. Only 2 of the 6 spiked substances, the ditert hydroxybenzyl alcohol and the bisphenol M, were detected as a concentration of 10 ppm. With a concentration of 1 ppm, only bisphenol M was detected. In an untargeted analysis, none of the other compounds would have been found. And as a reminder, the reporting limit in the BPOG protocol for organic substances is still less than 1 ppm. It's 100 ppb. The signals of oleamide and DHAP were found in the 10 ppm spike only due to a targeted search. Both compounds are well below the PS80 matrix and do not provide visible peaks in the BPC or UV. Without a targeted search for the mass tracks, those compounds would not have been identified. I had already mentioned the quantification of extractables. This compilation here is showing the response factors of known extractables in relation to Eganox 1010, bisphenol A, and the reverse ionizing compound of all listed substances. It is to illustrate how important it is to select the correct standard for semi-quantitative determination. The colors show the discrepancy between the true and the measured concentration if the corresponding compound in the first column would be quantified against Eganox 1010 BPA or the reverse ionizing compound by a semi-quantitative approach based on MS data. The green coloring represents a discrepancy factor up to a factor of 15, which is assumed to be acceptable compared to the others. Using the example of the ditert hydroxybenzyl alcohol, there's a big difference whether Eganox 1010 with a factor of 8 or BPA with a factor of 70 is used as quantification standard. And also for Ergaphos 168 oxide, that's the second last entry in the table, it's shown by the 2 lines that there's more than 1 mass track that can be used for quantification, and the colors highlight that the result depends not only on the quantification standard, but also on the mass track that is used for quantification. To sum up my key messages and considerations, standardization is beneficial in terms of standardizing expectations for supplier data, which leads to minimization of overall industry costs and acceleration of timelines. To get meaningful and comparable data, protocols have to be well defined, especially for analytical requirements. For most of our studies, no additional extractables were present in sodium chloride and PS80 profiles. The complexity of high salts and PS80 matrices decreases the sensitivity of instrument, and at the end, for an untargeted LCMS, the quantification of UV inactive substances is very complex, and there's a lack of confidence in the true value. So at the end, I want to thank all the people that have contributed to this presentation. Special thanks go to Jessica Shea and Paul Killian, Satish Kumar, Mohan Velu, and all the lab technicians that have been generated the data. Thank you also for your attention, and if you have any question that's not answered in the following Q&A session, please send an email to me to address that is shown here. I will take care that you will get an answer. Thank you.
Thank you, Dr. Haehn. Now we will begin the question and answer segment of the webinar, but once again, I would like to remind the audience that you can still send your questions in via the questions tab located directly below your webinar screen. So now our first question. Should the solutions be removed if no extractables are observed with that solution? Ekta, would you like to answer this first?
Sure. Yeah, so just because we don't see any extract in the solution does not mean that it should be removed, because it could be based on that particular one component that we don't see it, but also going back to how do we justify to our health authorities that the bag or the tubing or any of those single-use components are suitable for use. So if you don't see extractable, it's actually a good thing because then you can show that with the worst case, I don't see anything, so I can use my solutions, if they're less rigorous or compared to the solution that was tested.
Thank you. Our next question is, when performing extractables testing, do you pre-flush the filters? Saskia, would you like to answer this?
Yeah, sure. No, we do not pre-flush the filters. After the pre-treatment of the filters by autoclaving or gamma irradiation, the filters are used as they are and are extracted directly.
Thank you. And is there a risk assessment portion in USB 665? Again, Saskia?
No, there's not. The risk assessment portion was moved from the chapter 665 to 1665 because there was a considerable industrial resistance to 665 containing a mandatory risk evaluation matrix. I think that the industry would like to have the flexibility to use the matrices they have already developed and which they are currently using.
Thank you. Our next question. Can we remove PS80 from the solution list since it is difficult for analytical instruments? Ekta, would you like to answer this?
Yes. Not really. So I think my question is back to the lab people for analytics. The fact that PS80 is difficult is, are we using the right analytical techniques? I know we keep hearing this challenge, and the reality is it used to be tested before this protocol came out, so the suppliers used to test it, but we send them, when we paid for it. But my real question is, are we using the right analytical techniques? We use polysorbate in our processes, and we do have to measure to show that our product is stable. So we don't really use the, I don't want to say the excuse, but it is, it degrades, but we still are able to measure it and quantify it. So are we using the right analytical techniques, and, with all the advancements, are we moving in the right direction?
Thank you. Our next question. Do you ever submit the BPOG extractable data to support regulatory submissions in place of leachable data, realizing that extractable screening methods are not validated? Ekta, would you like to answer this first? And I'll come to you, Saskia.
Yes. Yeah, so it's not in lieu of the leachable. We are expected to submit extractable data as well as leachable data. So we could submit our internal generated data, or we have the option to submit data that we get from the suppliers.
Thank you. And Saskia, do you have anything to add?
No, as a supplier, we do not submit extractables data. We support the end users with the data, and they submit them to the further.
Thank you, then. I think, unfortunately, that's all the time we have for questions. But thank you, audience members, for the questions you sent in. And thank you, Saskia and Ekta, for your answers. But before I finish the webinar, I would like to ask our presenters, Ekta and Dr. Haehn, if they have any closing remarks. So, Ekta, do you have any closing remarks?
Just to say that if we really want to continue to move single-use forward, we need to continue to collaborate and work as an industry and find the solutions instead of trying to figure out what is the least, instead of creating roadblocks, let's find solutions, because end of the day, it's really for the patients. We are making drugs to help patients to improve lives, and we need to put in products out there that will help get the drugs faster to their patients but not cause any adverse events, and for suppliers when they're thinking about their products and trying to think about, just think about if it's something that a drug has to be used by someone you know, and your product is used in it, used to make that product, do you feel safe and comfortable that whatever your product is being used to make it, and it would be nice, it would be a safe product to deliver to someone that you know. Okay, so that's always something to keep in mind.
Thank you. And Saskia, do you have any closing remarks?
Yeah, I think in the last year the collaboration of the suppliers with the end users take us a lot more further, and I hope so that this will be in the future.
Thank you. We do actually have time for just one more question. So pick this question. Is this BPOG protocols also applicable for filters? Saskia, do you have anything you want to say about that one?
Yeah, as I have shown in my presentation, we performed the BPOG protocol on filters. Yeah, they are also applicable for filters.
Perfect then. So I would like to take this opportunity to thank our presenters. Once again, that was Ekta Mahajan from Genentech/Roche and Dr. Saskia Haehn, Laboratory Manager for Extractables and Leachables at Central Analytics of Merck, KGaA, Darmstadt, Germany, for sharing their knowledge with us. I would also like to remind our audience that you can view this and other webinars on demand by visiting biopharma-asia.com/on-demand-webinars. And if you are watching this on demand, then please feel free to send your questions over to me at stephen.edwards@biopharma-asia.com, and we'll get those answers sent back to you. Thank you, everyone. Have a great day.